Well, really, thank you for that lovely introduction, Guillaume. That was very sweet. And so today I'm going to be talking about my master's work in which I was examining predator-prey interactions and space use strategies into mesocarnivore species. And so starting off with the predator-prey side of things, these interactions have long been considered a key driver of evolutionary and ecological processes. And these interactions are not static, but rather they're like a game. Both sides have to weigh the risk and reward in making some move in a certain area or in a certain situation. And this decision can vary over space, with time, and even with the internal state of an individual. And so these interactions can scale up and impact population dynamics and potentially alter the community structure via trophic cascades. However, these predator-prey interaction studies have often focused on larger-bodied apex predators rather than those mid-tier mesocarnivore species. And so let's meet our players in this predator-prey spatial game. Today, I'm going to be focusing on two mesocarnivore species, bobcats and coyotes, and then an abundant prey species, white-tailed deer. And so starting off with our predators, the reason I focused on coyotes and bobcats is because they're two of the largest mesocarnivores that we will find in the state of Illinois. And given the removal of larger apex predators from this area, it's more likely that these two species will be able to fill that ecological role over other mesopredators. In general, mesocarnivores tend to act as forage and habitat generalists, and they're also able to persist in human-modified landscapes, which means they might be able to adapt and fill a role that those larger-bodied carnivores previously could not. When we specifically look at coyotes, we can see that they've greatly expanded their range beyond their origin in the open plains and deserts in the Southwest United States and are now found throughout the US. However, when we compare this to bobcats, bobcats have had a much more tumultuous history in the state. Their numbers decreased back in the mid-1900s. They were nearly extirpated from the state. However, their numbers have gradually increased, and they are now found often in the southern third of the state. And so here we can see this darker area at the bottom of Illinois. That's where bobcats are more likely to occur because they are considered a forest-dwelling species, and this is where most of the forests are located in Illinois. Now, moving on to our prey species, I focused on white-tailed deer because they are extremely important in terms of their management value, both their economic and ecological importance. And so they too have undergone this large range expansion, and they are now found all the way from South America up to Canada. And so many areas are experiencing a high abundance of deer, which can cause major losses in terms of forestry, agriculture, and even overforaging of certain habitats. And so it's really important to understand if coyotes and bobcats can fill that role of predator and impact white-tailed deer populations, or if other management tactics are necessary. And so one way that bobcats and coyotes could impact white-tailed deer populations is through this idea of direct predation effects. And this is when predators successfully kill prey at high enough levels to actually drive prey population dynamics. And so a classic example of this is the relationship between lynx and snowshoe hare. And so when we consider the example we've been focusing on, bobcats and coyotes have been shown to predate adult white-tailed deer. However, it's likely not at levels high enough to truly impact white-tailed deer populations. And instead, if they're going to have some direct impact on these prey populations, it'll likely be due to predation of fawns. And this is important because variable juvenile survival has been shown to drive population dynamics in other ungulate species. Additionally, I want to note that this risk of fawn predation has been shown to vary with landscape heterogeneity. Fawns will be at risk in some areas more than others. However, if they're not having a direct impact, these predators, I think they could be having some sort of indirect predation effect. And an indirect predation effect is when the mere risk of predation can cause prey to alter their morphology and even shift their behaviors. And some of those changes in behaviors can look like increasing their time being vigilant. It can also lead prey species to aggregate into herds, and prey can even alter their movement patterns and habitat selection. And so if you're spending all this time responding to predation risk, it follows that you could suffer some decrease to your overall fitness. And this is because if you're spending lots of time trying to avoid getting predated, you could be having less time to forage, which could lead to decreased reproductive success. And prey can also suffer increased stress levels or some combination of these factors. And so this too can go on to shape population dynamics. And so that leads us into my first chapter. And I had two questions I wanted to address. First, I wanted to determine which habitats have increased fawn predation risk on the landscape. And secondly, I wanted to determine some of the drivers of that increased fawn predation risk. And so what could be driving increased fawn risk on the landscape? So here we're just imagining we have a bunch of fawn kill sites on the landscape. Well, fawns could be at more risk in certain areas because does are spending a lot of time in those areas. It follows that wherever does are during that fawning season, fawns are likely not too far away. And so spending time in those habitats could lead to their fawns suffering an increased risk. Additionally, fawns could be at an increased risk of predation because predators are spending a lot of time in certain areas. Or it could be that predators are efficient or just really good at killing fawns in certain areas. So they might not be selecting that habitat, but they're just good at killing fawns in certain areas. And so here I've shown the predator relationships with these brown dashed lines. And this is because with the analyses I can't have a situation where a predator is both selecting a habitat and also more efficient in it. However, I do want to note that the predator and prey responses are not mutually exclusive. So it could be due to both a doe response and a predator response. And so here I'm going to be walking you through a conceptual framework for my first chapter. And we're going to start off with fawn kill sites. And so if I compare fawn kill sites to landscape availability, I can get at that first question. What are the riskiest landscapes for fawns? I can then compare fawn kill sites to doe locations to start getting at the drivers of increased fawn risk. And I can, for doe specifically, I can answer where are does especially at risk of fawn predation. Third, I can compare fawn kill sites to predator locations to determine where predators efficient or really successful at predating fawns. And finally, I can compare predator locations to the landscape availability to determine what landscape attributes predators are selecting and avoiding. And so the reason I have those two predator perspectives that you see is because I really want to tease apart if it's predator efficiency or selection driving increased risk. And so for the two models that are comparing to the landscape availability, I'm going to be using a resource selection function or an RSF. And then for the other two models, I will be using a latent selection difference function or an LSDF. And so here I've just popped up the names of what I'm going to be referring to these four different models as, and this will become important in the results because this is how I'll refer to them. So I have fawn kill sites to the landscape is going to be my landscape perspective. Fawn kill sites to does is going to be the prey perspective. Fawn kill sites to predator use is going to be that predator perspective. And finally, predator locations to the landscape will be predator selection. And so what exactly is an RSF? Well, let's imagine we have some hypothetical study area on the landscape with some level of landscape heterogeneity. We can have these ex-urban areas, which you can think of as the interface between rural and urban areas. They're not as densely populated as suburbs, but they're not completely rural either. We can also have cropland and even forested areas, just to name a few. And so within this study area, we can have some fawn kill sites on the landscape. Well, if we compare those fawn kill sites to a bunch of random points spread across the landscape, we can determine what landscape attributes are positively or negatively associated with fawn risk. And so building off of this, there we go. What is an LSDF? Well, we're going to think of it as an RSF, but instead of using random points, we can have another species act as our random points. And so this allows us to assess the selection difference between these two groups. So going off that same example, we're going to have our study area again with those fawn kill sites on the landscape. But this time we're going to be comparing them to doe locations on the landscape. And so here I wanted to note that does are not distributed randomly across the landscape. They too have their own selection and avoidance of certain resources. However, using an LSDF, I can actually quantify the selection difference between these two focal groups and the strength of that relationship. And so for my first chapter, my study took place in Jackson and Union counties here in Southern Illinois. Specifically, a lot of the fawn work took place in Giant City State Park, which I have outlined in blue here. And we also relied on the SIU Touch of Nature properties, which are two properties outlined in pink. And so data was collected from 2018 to 2023 by many hardworking grad students and technicians who went out and collared bobcats, coyotes, and adult white-tailed deer during the winter. And then fawns were found with foot searches from mid-May through the end of June during that peak in fawn births. And so since I was really interested in how does and predators would respond to that peak in fawning, I subsetted my GPS points to be from May 15th through July 15th. And so I ended up with 11 predators, 8 bobcats, and 3 coyotes, 54 does, and we ended up tracking 136 fawns and ended up with 23 fawn kill sites. And so here I want to note that in my models, I'm going to be treating bobcats and coyotes as a single predator. I'm not going to analyze them separately due to a limited sample size. And so once I had those fawn kill site locations on the landscape, I was able to generate a population level study area. And I did this using a 95% kernel density estimation with those fawn kill sites. And I'm going to just show you what that study area looks like. So here we have those fawn kill sites in red, and the outline, which is kind of purple on this screen, is the study area I ended up with. And it ended up being 86 square kilometers and consisted of 69% forest, 7% cropland, and 6% exurban areas. And so once I had that study area, I retained predators and does with approximately 100 GPS locations in this study area to satisfy the assumptions of the LSDF. And I say approximately 100 because one bobcat had about 97 locations. And that's the sample size that you saw on the previous slide. And so to determine the relationship between fawn predation risk and landscape heterogeneity, I used many available remote sensing products. And I'm going to show an example of what one of those products look like. So here I have distance to road for my study area of interest, and I have those fawn kill sites plotted again. And here what I would do is I would generate a buffer around each location and extract the mean distance to road for that buffer size. And so I did this with various spatial covariates. I looked at 6 different land cover classes: distance to road and water, canopy height, elevation, and terrain ruggedness. And for my buffers, I wanted to test 10 different spatial buffers because I wanted to determine what spatial scale had the greatest impact on fawn risk. And so let's see an example of what one of those buffers looks like. So here we'll focus on this fawn kill site location outlined in brown. And these buffers ranged all the way from 50 meters up to 500 meters. And so I tested these 10 different spatial buffers across my 3 models which contained fawn kill sites. And I found that the top model was the 100-meter buffer. And so before we dive into the results, I just want to orient everyone because it'll end up being a fair amount to look at. And so on the y-axis, I have my spatial covariate of interest. And specifically, I'm focusing on covariates that were significant or marginally significant from the landscape and prey perspectives. And this is because I was most interested in the risk of predation from the prey's point of view due to the high cost that predation carries. On the x-axis, I'm going to be having values for my coefficient estimates and 90% confidence intervals. And so what you're essentially going to look for is each model is going to have some point estimate along with an error bar. You're going to look first, does that error bar overlap this dotted line of 0? If not, is it positive or negative? And so moving on to question 1, where are fawns at an increased risk on the landscape or that landscape perspective? Well, fawns were at an increased risk of predation further from roads and in greater proportions of crop, forest crop edge, exurban, and other land cover types. I also found that fawn kill sites did not differ from landscape availability for canopy height. And so here we can see that, let me get my laser pointer up real quick. For canopy height, that error bar overlaps 0 while the rest are positive. Now moving on to the second question, getting to those drivers of increased fawn risk, we're going to focus on the prey perspective first. And so here we're going to be looking for a pink diamond to pop up. And so I found that fawns were predated at a rate similar to doe use further from roads and in greater proportions of exurban. I also found that fawns were at an increased risk relative to doe use for crop. And this was kind of trending for edge and other land covers. However, it was not significant at the 90% level. Finally, I found that fawns were safer relative to doe use in higher canopies. Now, when considering the predator side of things and what might be driving fawn risk, we're really trying to tease apart that predator efficiency versus selection. And so here I'm going to be focusing on predator selection first, which will be this yellow circle. And I found that for all of these covariates, predators either did not select or even avoided these resources. So most of these error bars overlap 0, except for the other land cover type, which they actually avoided. And instead, it seems that relative to predator use, more fawns were predated. And so this suggests that predators were especially successful or more efficient at killing fawns in certain areas rather than others. Again, the only covariate this did not occur for was canopy height, which overlaps 0, and the other predator perspectives are positive coefficients. And so let me get rid of my laser pointer. For the drivers of increased fawn predation risk, it seems it is largely driven by predator efficiency and partly by doe selection of risky habitats, and not at all by predator selection of habitats where fawns are at an increased risk. And so I really want to focus on this idea of why might does be spending more time in an area where their fawn is at increased risk? Well, in terms of road use, I think it could be a trade-off between fawn risk and doe risk. And that's because, as I mentioned earlier, adult white-tailed deer can be predated by coyotes and bobcats. However, they're much more likely to die due to a vehicle collision. And so does might be selecting areas further from main roads. And by doing that, they could be potentially putting their fawns at a greater risk of predation. And so for them, it could be this trade-off between their survival and the survival of their offspring. When considering ex-urban areas, I think it could be a trade-off between fawn risk and forage. And so I'm going to walk through an example of what could be playing out on the landscape here. And so here we have some semi-rare ex-urban area, which has this nice big lawn, a house, and even a few bird feeders. And these areas can actually be really great habitat for does because they can offer access to some high quality forage. Additionally, does may also think that they can be less vigilant due to this idea of the human shield effect. And this is where predators tend to avoid areas where humans are more than prey do. And so when the doe is considering her fawn, she might think her fawn is safer in these ex-urban areas. However, for the predator, the fawn is at increased risk. And this is likely because bobcats and coyotes don't venture into these ex-urban areas often, but when they do, it's likely an easy pursuit given the openness of the habitat and lack of hiding cover for the fawn. This leads to the fawn likely ending up predated and overall at a greater risk in these ex-urban areas. Interestingly, I did find that does may be aware of the increased risk of fawn predation relative to landscape availability in certain areas, and specifically we're going to be focusing on croplands. And so croplands and pastures tend to occur on more productive soils than forest, and they too can offer access to forage for does. However, maybe they're sensing some cue and they're able to be more vigilant and overall decrease their time in these risky areas. And instead, they might reallocate that time to safer areas, such as higher canopy heights, where their fawn is at a lower risk of predation. And so for my first chapter, my first question, determining which habitats have increased fawn predation risk, I found that fawns were at an increased risk further from roads and in greater proportions of ex-urban, crop, forest crop edge, and other land covers. For my second question, looking at some of the drivers of increased fawn risk, it seems it's largely driven by predator efficiency and maybe partially by doe selection of some risky areas. And so a question I was really left with after this chapter was this idea of how is predator efficiency structured? And what I mean by that is, are all predators responding in a similar manner to fawns, or are some just really good at killing fawns on the landscape and others may not be responding so strongly? And the reason I was interested in this is because past work in this lab has actually found that bobcats and particularly coyotes can exhibit substantial individual variation in their spatial responses to fawns. And so here I'm going to pop up some results from that past study. So here, hopefully everyone can see that. When we're looking at bobcats, out of the 8 bobcats we looked at, only 2 displayed some sort of selection for fawns. While when we look at the 16 coyotes we had, about half or 8 coyotes displayed some selection towards fawns. And so for my second chapter, I really wanted to build off of this idea of individual variation and quantify that in some movement and space use behaviors, specifically for coyotes. And so before we get into my second chapter, I want to take a step back and consider, why do we care about the steps an animal takes during its lifetime? And so here, I'm showing an example of what GPS data might look like for us. So here, each point represents a GPS location, and the line in between represents some trajectory of an animal. And so when we look at this scale, we might not think much is going on, like it just looks like some points on a map. However, when we zoom out a bit, we can see this was actually a really important moment in this animal's lifetime because this directed movement was when this animal was escaping a predator. And we can compare that to these more tortuous movements or really curvy movements. And this is a time when an animal is searching for food. Zooming out even further, we can see that this middle trajectory just represents a small piece of this animal's lifetime track, all the way from birth until death. And so these lifetime movement trajectories can vary across space, through time, between individuals, and even among populations. And so some of the variation in these movement trajectories can be attributed to external factors, such as the spatiotemporal distribution of resources. Are they really clumped on the landscape, or are they randomly spread? Variation can also be due to the presence of social or cultural learning, and even the level of human modification present. Additionally, variation in movement can be attributed to internal factors, such as the life stage or age of an animal, sex, or even the broad-scale movement strategy employed, such as residents versus migrants. And so we can attribute variation in movement to all these different factors. But even after doing that, variation can still be attributed to an individual. And this is due to differences, non-reversible differences in genetics or developmental history. And these individual level differences can have important impacts and real consequences for an animal, because it could be the difference in obtaining certain resources. It could also make the difference in finding a mate and reproducing, surviving, and it can even offer some animals a way to find shelter from land use and climate change. And so how do we quantify individual variation? We can account for all these different factors, but how do we determine how different are individuals from each other? Well, one way is to use behavioral reaction norms, or BRNs, as I'll be referring to them. And so these models for my stats folks are just often generalized linear models with a random intercept and slope by individual. And so from these models, we can extract various components that describe some aspect of individual variation, such as repeatability or the consistency of a behavior. And so here's the equation for it, but essentially you just need to know that it's the ratio of variation among individuals divided by the total variation measured by the model. And so another thing to note about repeatability is that it's on a scale from 0 to 1, and low values represent low consistency of behavior. We can also quantify personality, or the average behavior of an individual, and plasticity, which represents how behavior varies across some gradient of interest. So here I'm going to pop up an example of what I mean by this. So here we have behavior X on the Y-axis and some environmental gradient on the X-axis. And those three lines represent a different individual. And so here that we can see that these three individuals differ in their personality or their individual level intercepts. Well, two of these individuals actually have the same plasticity or the same slope. And so we can add more and more behaviors to this BRN framework and expand it to understand if a behavioral syndrome is present. And this is an among individual correlation between behaviors. So you can do this with as many behaviors as you want, but we're going to focus on just two behaviors. So here we're looking at behavior X and Y, and we're going to focus on those same three individuals again. So here we can see that there's some positive correlation between behavior X and behavior Y across individuals. And so the BRN framework has been applied to some movement and space use behaviors. It's still relatively new, but we have learned more about foraging and habitat selection. However, we truly don't understand how this variation may be structured across spatial scales. And so I want to walk through an example of what I might mean by this. So here we have two populations, population A and population B. And in each of these, we're measuring behavior X across the same environmental gradient. And for both of these populations, we tracked 5 individuals each, and we measured their behavior multiple times across this gradient to produce these lines. Well, if we average those individual level behaviors up to the population level, we see that these two populations actually exhibit opposite responses in these behaviors. Population A tends to increase the expression of behavior X, while population B tends to decrease the expression of behavior X. And so what if we had not accounted for scale or the population level context? So here, I have those same 5 individuals from those populations, but instead I'm ignoring what population they come from and I'm just lumping them all together. Well, if we average those behaviors, we would see something like this. And here we would conclude that behavior X is pretty consistent, or it doesn't really change as the gradient changes, which can have some real impacts on determining how behavior may change across an environment. And so I think it's easy to see that this point may seem a bit trivial. However, in the era of big data, it's becoming easier and easier to get GPS locations and behavioral observations from animals across the US, across the world, and it's getting easier to ignore the scale at which that behavior may actually matter and just lumping things together and canceling out behaviors that may truly be interesting and have some real consequences. And so for my second chapter, I wanted to address 3 main questions. First, I wanted to attribute variation in space use behaviors to external and internal factors. I then wanted to quantify if individual variation was present within populations. And third, I wanted to determine if behavioral variation was consistent across broader spatial scales. And so for my second chapter, I'm going to be walking you through another conceptual framework. And so to get at my 3 questions, I'm going to be using data from 3 disparate populations of coyotes across the state of Illinois. So one population is primarily forested, which is green. One population is agricultural in blue. and one population is urban coyotes in orange. And so from these three populations, I'm able to decompose their GPS data into two behaviors of interest. I'm focusing on one movement behavior and one space use behavior. And here you can see their names, speed and RSS. Don't worry about that right now. I'm going to go into the methods, what those behaviors actually are. And so using the model we see at the bottom here, I can attribute variation in these two behaviors to various fixed effects. And here I'm going to be focusing on some external and internal factors that could be driving some of that variation in these two behaviors. After attributing that variation to my fixed effects, we can take a look at our random effects. So here we have two plots, and we have RSS and speed on the y-axis, and then my gradient of interest, which is proportion of forest on the x-axis. And so here, each line represents a different individual from their respective populations. We can then, and so that gets at that second question, quantifying if individual variation is present. To get at the third question, we can then average those behaviors and see what does behavior look like at the population level and how consistent are those behaviors across populations. There we go. And so to capture this shift from forest to ag to urban, I'm going to be using data from 3 sites across Illinois. We've already met the first site, but here I'm going to be referring to it as Southern Illinois throughout, and this is our forested site. For the agricultural site, or Central Illinois, all trapping took place in Shelby and Moultrie counties. And this area is dominated by agriculture with the forest restricted to the edges of Lake Shelbyville, which you can kind of see here. And then for the urban site, we're actually using data from the Urban Coyote Project out of Ohio State University. And their trapping took place in Cook, Kane, and DuPage counties. And this area is the Chicago metropolitan area, or what I'll be referring to as the CMA. And so here we can actually see a ground level example of what these different landscapes might look like for a coyote. And so on the left, we have an example of what a coyote might encounter in Southern Illinois. In the middle, we have what a coyote might encounter in Central Illinois, those open areas. And finally, we have a street in downtown Chicago, which we did have coyotes down in the middle of Chicago. And so coyotes in Southern and Central Illinois were trapped from 2018 to 2023, while coyotes in the CMA were trapped from 2008 to 2019. And during the workup process, we collected various metrics such as age in terms of juveniles versus adults, sex, and GPS frequency. And so the fixed intervals, or the rate at which a GPS location was taken, varied greatly across years and also across study areas, with Southern and Central Illinois taking a GPS location every 1.5 or 3 hours, while the CMA initially took a location every 7.25 hours and then adjusted that to anywhere between 3 hours to 15 minutes. And so this difference in fixed interval becomes important when I'm trying to calculate something like speed, which is distance divided by time. And if that time differs across those study areas, we have to find some way to account for that difference. I also subsetted my GPS locations to 2 seasons of interest, which I defined as winter, which is December 15th through February 15th, and then fawning, which has the same definition as the first chapter, May 15th through July 15th. And the reason I was interested in these 2 seasons is because I wanted to see if behavior would shift as the amount of food availability increased on the landscape from winter to fawning. And so to get these repeated behavioral measures for the BRNs, I used what's called a rolling window. And so let's go through an example of what that actually looks like. So here we're considering May 2023, and we're going to start off at May 15th at the start of the fawning season, and we're going to count out 5 days to May 19th. And I was able to average my 2 behaviors during this 5-window period for each coyote, and I've called this behavior on days 1 through 5. We can then shift that window by a day and get behavior for days 2 through 6, 3 through 7, and so on until we reach the end of the window. And so as I was doing these rolling windows, I also calculated my gradient of interest, which is that proportion of forest. And so I calculated this using a 95% minimum convex polygon during those 5-day periods and just calculated the proportion of forest available for each coyote. Moving on to my behaviors, we're going to focus on the movement behavior first of speed. And so to calculate speed, I used autocorrelated movement models in our package CTMN. And the reason I did this is because I can actually calculate a scale insensitive speed. And what I mean by that is it can actually account for biases due to sampling frequency, which I mentioned very greatly across the site, so it accounts for that. It can also account for differences in animal tortuosity, or how much the animal is turning as it travels along its path. And it can account for the amount of GPS error associated with a specific color type. And so from these models, I extracted speed in meters per second for these 5-day rolling windows for each coyote season, if that top model had autocorrelation associated with it. Otherwise, the window was omitted. And so here, there was no way to really show my speed model, so I wanted to show some examples of what speed might look like on the landscape. So we have a coyote in the upper left that is in the forested region or southern Illinois. To the right of that, we could see a coyote moving through central Illinois, that agricultural region. And finally, at the bottom, we could see how a coyote might move through Chicago. For my space use behavior, I looked at resource selection specialization. I'm going to be calling that RSS or specialization from now on because it's a mouthful. And it's a simple metric that can describe the magnitude of an individual's average response to given resources, regardless of directionality. And don't worry, we're going to build up to what that looks like. So to quantify specialization, we first had to quantify resource selection. And I did that using Iblev's electivity index, or EIRT. And this value ranges from -1, which represents total avoidance of a resource, to +1, which represents total selection of that resource. And here, 0 represents no resource selection or avoidance. And so to get from selection to specialization, I'm going to walk through an example. So here we have 4 different individuals, and I've plotted, there we go, their electivity indices for 2 resources, resource 1 and resource 2. Anything below the 0 line represents avoidance of a resource relative to its availability, while anything above represents selection of a resource relative to availability. And so here, when we look at, say, the green individual, we can see it's selecting both resource 1 and resource 2. However, when we look at that reddish pink individual, we see it's strongly avoiding resource 1 and pretty strongly selecting resource 2. Well, if we take the absolute values of these indices, we'll end up with something like this, which we can then average by individual to get our specialization values. And so here we see that that red individual who was really strongly avoiding and really strongly selecting resources ended up being our most specialized. And so the nice thing about this RSS metric is that given that we took the absolute value, it ranges from 0 to 1, with 0 representing a resource generalist to 1 representing a resource specialist. And so again, similar to speed, I averaged RSS for those 5-day windows for each coyote season. And then once I had those 2 behaviors extracted, I tested various combinations of random effects. And I found that the top model included a random intercept for year and individual, along with a random slope by individual for the proportion of forest experienced. And so for the first question, attributing variation to external and internal factors, I'm going to be showing you 2 plots with RSS on the left and speed on the right. And here we're considering the external factors of site, season, sex, and age, which are all categorical variables. And then forest is that continuous variable, the proportion of forest. And so here the reference category or intercept for this model is an adult female coyote from Chicago during the fawning season. And I know that's a lot, so I'm going to try and keep you oriented about what these results actually mean as we go throughout. So focusing on the external factors first, I found that coyotes in central Illinois increased their specialization, but decreased their speed relative to CMA coyotes. Additionally, I found that southern Illinois did not differ in terms of specialization or speed relative to CMA coyotes. I also found that coyotes in winter were more specialized than coyotes during fawning. And I found that coyotes moved slower as the amount of forest they experienced increased. When looking at some of the internal factors, I found that juveniles were more specialized and slower when compared to adults. And I found that males were more specialized than females, but they did not differ in terms of speed. And so after attributing variation in RSS and speed to those various external and internal factors, I still found substantial individual variation or differences between individuals. And so we'll first consider repeatability. For RSS, I actually found a relatively low repeatability at 0.15. And what this value means is that 15% of the variation in RSS can be attributed to consistent individual differences. And so this low repeatability could be the result of individuals behaving inconsistently through time or not behaving, or behaving too similarly to each other. For speed, I actually found a moderate to relatively high repeatability at 0.51. And again, this value means that 51% of the variation in speed can be attributed to consistent individual differences. And so moving on to personality and plasticity, I'm going to be showing a similar figure that we saw in the conceptual part of the talk. And so here we have RSS on the top and speed on the bottom, and both of those behaviors are on the y-axis. And on the x-axis, I have the individual level of forest availability. The reason I call it individual level is because not all coyotes experienced a full range of forest. We can see that coyotes, specifically from the CMA, experienced very low levels of forest, and that's on the far left-hand side. They have really short lines, and that's because they didn't experience the full range of forest. And so for personality, or those individual level intercepts, I found substantial variation for both RSS and speed. And then when we look at plasticity, or those individual level slopes, I found that RSS exhibited some complex slope patterns, with some individuals decreasing specialization, some increasing, and some exhibiting almost no change as the amount of forest increased. When looking at speed, I actually found that most individuals did tend to decrease their speed as the amount of forest increased. And then finally, when comparing or looking at a correlation between these behaviors, I found minimal evidence of a correlation or some behavioral syndrome between these behaviors. And so these findings suggest that individuals do exhibit variable personalities and behavioral responses. However, it's probably not to the extent that some folks like to think. So this is out of an article out of San Francisco, where a woman likes to assign personalities to different coyotes. And so slightly different, but that's all right. All right, moving on to the third question, determining if variation is consistent across populations. We're going to be looking at those individual level results again. And if we average those by site, we'll end up with something like this. And so here, I found that personality did differ across sites, or those site level intercepts did differ. And then for plasticity, again, I found RSS had variable responses, with Southern and Central Illinois exhibiting opposite responses, and then the CMA appearing less flexible in specialization relative to forest. And then for speed, I actually found a similar decrease across all sites. Those are actually very similar slopes. And so what does this all mean? I know that was a lot, so let's break down some of those results. However, given the time I have today, I don't want to keep you here all day. We're going to focus on one fixed effect result, which is, why are Central Illinois coyotes more specialized and slower relative to Chicago coyotes? So considering specialization first, coyotes in the CMA and Southern Illinois sites have a more even distribution of forest to open to agricultural areas when we compare that to Central Illinois, which is really dominated by agriculture at 79%. So I think Central Illinois coyotes may be responding to this uneven distribution of resources by really strongly selecting forest when there isn't much available on the landscape. And so if we walk through an example of that, let's say a coyote had 3% forest available to it. It might be strongly selecting that resource given its limited availability compared to if there was, say, 50% forest available. In terms of speed, I think CMA coyotes could be moving faster than Central Illinois coyotes due to the extreme urbanization of Chicago. So urban landscapes often provide these high quality but highly clumped resources such as anthropogenic food or shelter. However, the distance between these resources are often quite large. And so many species respond to this clumped resource distribution by constricting their home range and taking advantage of just a few high quality patches. However, coyotes have been found to be one of a few species that actually expands its home range in urban areas. And this suggests that urban coyotes may have to move faster between patches in order to meet their energetic demands. And so again, let's look at an example of what speed might be looking like in Chicago and Central Illinois. So here on your left, I have a Chicago and Central Illinois, and we can see it keeps making these movements where it'll go up, but it often returns to this rectangular patch, which I looked into, and it's a railroad yard. And then when we compare that to Central Illinois, the agricultural coyote doesn't make as much back and forth movements between presumed patches, and instead it seems to have a more directed movement. When moving on to questions 2 and 3 and those variable specialization responses, I found opposite responses for Southern and Central Illinois. And the reason I think I might find this decrease in specialization for the agricultural area and then an increase for the forested area is because forests differ in value across these two sites. And so in Central Illinois, coyotes actually face really strong hunting pressure and persecution compared to Southern Illinois. There's a big trapping community up there. And so when there's little forest available, those agricultural coyotes, or that blue line, may be more specialized because they're strongly selecting forest and avoiding those more open or developed areas as some form of refuge from hunters. And then for Chicago, it appears that they're actually less flexible in their behavior, or in their plasticity, excuse me, at the site level. However, I think that's driven by the pattern getting canceled out based on individuals. So I'm going to pop up my laser pointer again. So here we can see individuals that experienced roughly 0 to 50% forest availability tended to decrease their specialization for Chicago. However, those that experienced greater than 50% tended to increase their specialization. It's obviously not perfect, but I think that's why they may appear less plastic. When looking at speed, I actually found it was a relatively consistent behavior. I found a similar decrease in speed as the amount of forest available increased across all sites. And so this suggests that speed could be more consistent than specialization. And so why might coyotes be decreasing their speed as the amount of forest increases? Well, it could be they want to spend more time in areas with increased cover and this greater perceived safety. Additionally, they could be moving slower because forests are just really hard to move through. If you went out and tried to huck it in the woods right now, you'd run into a bunch of invasive plants, fallen logs, and there's no roads to walk along, so you're probably going to be slowed down. And so I really want to hammer this point home of what if scale was not accounted for. So here are those same figures we've been seeing this whole time with the individual lines averaged up to the site level. Well, for specialization, we would have found something like this if we had ignored the difference across populations. We would have concluded that specialization was pretty consistent as the amount of forest increased. For speed, however, we would have came to a similar conclusion. Speed appeared to decrease as the amount of forest available increased. And so it really does depend on your behavior of interest that you're considering. And so for my second chapter, for the first question, attributing variation in space use behaviors to external and internal factors, I found that site, season, age, sex, and forest availability all accounted for some of the variation in speed and specialization. And even after accounting for this, I found that there was significant among individual variability in these two behaviors. And then for my third question, determining if behavioral variation was consistent across broader spatial scales, I found that consistency really depends on your behavior of interest. Again, I found speed was much more consistent than specialization. And so what I really want to do today, though, is leave you with one final question. What does the existence of individual variation in space use behaviors actually tell us? Well, not much yet. And this is because most studies quantifying individual variation in behavior fail to determine if these differences in behavior actually matter for these animals. And so we have to determine if variation in space use strategies are related to things such as variable reproductive success and survival, which I'm currently looking at slightly for my second chapter. And so given that movement is a primary mechanism in which animals interact with their environment, I think there will be some difference in fitness. And this can have some important implications for the adaptive potential of varying space use strategies. And it can also help explain why different space use strategies are maintained within populations. And so in order to do this, future work will have to consider multiple different movement and space use behaviors, along with various environmental gradients. And so those gradients don't just have to be the physical landscape, but it could be a foraging gradient, predation gradient, and so on. And so with that, I would like to thank the SIU IDNR DEER project and the SIU Graduate School for funding my master's work. I would also like to thank the IDNR, SIU, and the Army Corps of Engineers, along with private landowners for access to field sites. I also have to thank all the technicians who worked on the adult deer, predator, and fawn projects, because without them, I wouldn't have had a master's to work on. I also want to thank Dr. Stanley Gert and Hans Ellington for loaning me the Chicago data. I also want to thank Drs. Clay Nielsen and Peter Schlichting for serving on my committee and providing feedback on my proposal earlier on. I want to thank Guillaume. You've been an awesome mentor this whole time, and I aspire to be someone like you in academia. And finally, I want to thank my friends in the Spatial Wildlife Ecology Lab and the CWRL, who made sure I had lots of fun while I was in Southern Illinois. I want to thank my parents who came out here today for just supporting me throughout my life and career. And finally, I have to thank Jake and our dogs for keeping me sane in Pennsylvania these last 6 months as I was finishing up my master's and making sure I got outside some. And so with that, I would be happy to take any questions.